Welcome back. Now we will start with the sessions where we will have short talks. And our first presenter is Lei Zhang from the University of Minnesota. Hi, Lei. Can you?
Yes. Can you see my slides?
Yeah, we can see your slides. Yeah.
Okay, perfect. Okay, thank you. Hi, everyone. My name is Lei Zhang, a postdoc research associate from Dr. Paul Robbins' lab at the University of Minnesota. My research mainly focuses on studying the mechanism of aging and developing therapeutic interventions against aging. Today, I'm happy to talk about one of my several drug discovery projects targeting aging and senescence, which is to inhibit NF-κB signaling with a small molecule to improve health span in aged mice. So NF-κB is an evolutionary conserved transcription factor in many species. As you can see, the NF-κB system is basically a bow tie structure stimulated by different stress, damage, or danger signals. It coordinates different cell responses to protect, repair, or grow. Under normal conditions, its activation is vital for many physiological functions, but its abnormal activation can cause many age-related diseases and aging itself. And more importantly, NF-κB overactivation is not only associated with aging, but also drives aging, also interacts with many other pro-aging and longevity pathways and factors. All of these make it a promising target for healthy aging interventions. We have been studying different components of the NF-κB system and their biological effects using both genetic and pharmacological methods. For example, genetic reduction of P65 delays the onset of aging symptoms in mice. Inhibiting ATM, either genetically or pharmacologically, can reduce NF-κB activity and thus suppress aging symptoms. We are currently working on NEMO mutants and knockout of TNF receptors to see their effects on NF-κB signaling and aging. So the focus of my presentation today is the small molecule SR12343 that can inhibit NF-κB activation by disrupting the IKK beta and NEMO association. We studied this project with virtual drug screening. First, the X-ray structure of the NEMO IKK beta complex was used to extract the chemical features essential in the protein-protein interaction. A structure-based pharmacophore was generated using Ligand Scout software. It consists of hydrophobic groups, hydrogen bond donors, hydrogen bond acceptors, etc. Then this pharmacophore model was used to screen a zinc drug-like database containing about 13.5 million compounds. After several filtering steps, eventually 3 hit compounds were selected for testing, and one compound can inhibit NF-κB at higher concentrations. To identify compounds with higher biological activity, the zinc database was screened for structure-similar compounds, and 15 analogs were identified and tested. Among these, zinc 5 turned out to be the most potent one, but it is extremely unstable even in cell culture media, probably due to ether bond shown here. So to improve bioactivity and stability, SAR studies were performed. More than 100 small molecules were designed, synthesized, tested, redesigned, resynthesized, retested, and among these, SR12343 was found to be the best analog. And we further validate that SR12343 can indeed inhibit NF-κB activation, and it selectively targets NEMO IKK beta interaction other than NEMO IKK alpha. Also, it has better stability in vivo after injection into mice. We have studied the therapeutic effects of this compound on 2 mouse models, acute inflammation and muscular dystrophy, and got very positive results. So given the important role of FKB in aging, we also wanted to know the therapeutic potential of this compound in aging models. So we used mouse and human cell models, 2 progeria mouse models, and 1 naturally aging mouse model. In all of these models, NF-κB has already been shown to be hyperactivated. First, we use oxidative stress to make senescent mouse cells and genetic stress to make senescent human cells. As you can see, SR12343 can reduce beta-gal activity in both cell models without causing cytotoxicity. It also reduces the expression of key senescence and the SASP factors like P16, P21, TNF-alpha, and MCP1. So encouraged by the in vitro results, we further test this compound in ERCC1 progeria mouse model with 3 injections per week for 6 weeks. As Dr. Gian Poyamakers described earlier today in his talk, these mice are deficient in DNA repair, thus age rapidly and recapitulate most aging features. So health assessment was done to evaluate the aging symptoms, and higher scores indicate more severe aging symptoms. The treated mice showed delayed and less severe aging symptoms. This is true for both male and female mice. As an example, the photo shows the dystonia, a movement disorder with involuntary muscle contractions, was significantly improved in treated mice. So then we analyzed the different tissues of these mice. In the liver, SR123 can reduce key cellular senescence markers like beta-gal activity, as well as P16, P21 at both transcriptional and translational levels. In the muscle, the drug treatment not only reduced the senescence, but also improved multiple muscle pathologies, including muscle fibrosis, increased muscle regeneration potential, and decreased inflammatory infiltration. Z24 knockout mice are another progeria mouse model. Z24 deficiency leads to unprocessed prelamin A and nuclear disorganization in cells, thus shorten the lifespan and accelerate aging phenotypes in mice. After treating deficient Z24 cells with this compound, there are less cellular senescence, less SASP factors, and less DNA damage. And muscle stem cells showed improved differentiation potential. Also, there are less cells with abnormal nuclear blebbing caused by Z24 deficiency. Then we test this compound in Z24 knockout mice with 3 injections per week for 10 weeks. The treatment reduces the level of senescence in skeletal muscle and heart tissues, consistent with the results in ERCC1 minus delta mice. SR12343 also improves muscle pathologies. There are less muscle fibrosis, more muscle stem cells, less CD68 positive macrophages in the skeletal muscle of treated mice. So last, we test this NF-κB inhibitor in wild-type old mice at about 2 years old as a later life intervention. Mice are treated with 3 injections every week for 17 weeks. In the liver, the drug treatment significantly reduced different senescence and SASP factors, as well as NF-κB activation, evidenced by phospho-P16 65. In the muscle, the treatment also significantly reduced different senescence and SASP factors. We are currently doing the histological analysis on the muscle tissues to see if any improvement on muscle pathologies. So in summary, we have shown that pharmacologically inhibiting IKK and NF-κB activation not only can suppress inflammation, but also can reduce senescence and SASP factors, improve health span, attenuate muscle pathologies. These results suggest that IKK and NF-κB signaling is a promising target for healthy aging interventions. And SR12343 may have potential for extending health span and treating age-related diseases. And by the way, we have been conducting further SAR studies to find more effective SR12343 analogs to improve oral bioavailability. And we already have identified several better ones, and we'll publish the results in the future. So with that, I would like to thank organizers of the meeting, my amazing collaborators from different institutes, and especially my advisor, Dr. Paul Robbins. And also thank you all for your time and attention.
Thank you very much for your presentation. We have some questions on Slack, and maybe we can just summarize them as one question. So the questions are from Morten and Garrick Makartichan. Have you tested your drug in old wild-type mice, for example, regarding behavior physiology, and in general, how wild-type mice behave after treatment with your lead compound?
Yeah, we did the health assessment on the wild-type old mice, and we have the data. They show positive results as the results in ERC1 mouse data mouse model. Due to the time limits, I didn't show the data over here. Thank you.
Okay, thanks. And there are some more questions on Slack, and you can just go online on Slack to answer them. Thank you very much for joining us today, and we will